This study is really interesting for the development of new therapies for cardiac disease. What happened in this project is we took samples from patients at the University of Kentucky that were donated for research, and we collaborated with a group at the University of Massachusetts Medical School, and they produced pictures of cardiac thick filaments at the level of individual molecules. So we got what's called a cryo-electron microscopy structure of the thick filaments that make up cardiac muscle. The heart's made up of billions of cells. Cells are made up of structures called sarcomeres, and within sarcomeres there are things called thick filaments. And so we're looking at the thick filaments that come from these cardiac cells. Each filament is made up of roughly 600 molecules with a really complicated structure. People have been trying to work out the structure of these filaments for 60 years. We knew quite a lot about how the molecules were arranged. We knew roughly how many of them there were. We knew that they were arranged in crowns of 3 pairs. This paper just gives an unsurpassed detail, and down to the level of like individual molecules, we can see that these 3 crowns are actually different. And that's interesting because it tells us something about how the cardiac muscle is turned on when your heart beats. And we can also see how some of the accessory proteins, like one in particular called myosin-binding protein C, how they sit in the structure. And that's a protein that often is a source of disease of people who've inherited a myopathy. And so trying to understand how that protein sits in the structure, that's really giving us a new level of information about how the molecules are arranged in the heart. This research is important for people in Kentucky because we are one of the worst states in terms of heart disease, and we desperately need better therapies. And so hopefully this work is part of many pieces of research being done at the university and across the state that are trying to help come up with better therapies for heart disease. One of the things that I'm most proud of about this work is that it comes out of the Gill Cardiovascular Biorepository. We call this the Biobank, and we started it in 2008. So now we've built a really big resource, maybe 15,000 samples from almost 500 people. And we share these samples with research groups around the world. This study that's just coming out in Nature is one of the collaborations that comes out of that bank. If you get a paper published in Nature, it means that a wide range of scientists think that this work is of importance to everybody, not just people who specialize in that particular area. I think the most important thing about this study in the sort of real hardcore science is that we know the molecular structure of cardiac thick filaments, and we can use that information to try and design better therapies.